Dear students, uh, we have been discussing about HIV and AIDS in the previous lectures. Today we will continue with that. In the previous lecture, we discussed about the structure of HIV, in which we discussed about the envelope proteins, about the GP120 and GP41. Now let's move inside this virus. As you see here, inside the virus, inside the lipid bubble, there are other types of proteins as well. The first protein which is there, that's just like I have made it like an M alphabet. So this M for matrix. These proteins are called matrix proteins. So these are the matrix proteins. These proteins are actually formed in order to support the GP41. So these are the supporting proteins which is called the matrix proteins, and they are P17. P for proteins, and 17 actually is their molecular weights. So this is this matrix protein. And now when we move inside again, we will see now what are the proteins of which the capsid is made up of. The capsid is made up of another type of proteins which are called P24. So these are P24 proteins. P24, P24. So the capsid of this virus is made up of P24 proteins. The matrix is P17 and the capsid is P24. Inside this there is a dimer of RNA, and it also carries with it three important enzymes which we can remember just like RIP, reverse transcriptase, integrase and protease. So now let us see how the virus infects the host cells, how it attached with the CD4 positive receptors. Now let's see how it attaches these receptors. For example, let's make a macrophage. This is a macrophage. It is a CD4 positive cell. We know it. Uh, this macrophage definitely will have which receptor? CD4 positive receptor. So here, suppose this is the CD4 positive receptor. So this is the CD4 positive receptor of macrophage. Another cells which we have discussed in the previous lecture, which has CD4 positive receptors, are the monocytes, helper T cells, microglia, and then all those cells we have discussed in the previous class. Now, what actually happens? Now, suppose this is the virus. This is the virus. It has this protein GP41 here, and the attaching protein, that is this GP120. So it will bind with the CD4 positive with GP120, and this is the CD4 positive receptor. CD4 positive receptor. Now this virus will bind with the CD4 positive receptor by the help of GP120. As I already told you that it is the GP120 which helps us in the primary attachment, primary binding with the CD4 positive. तो इसका मतलब हो गया कि GP120 की बदौलत ही ये पहले बाइंड करता है CD4 पॉजिटिव रिसेप्टर के साथ. फॉर एग्जांपल हमने एक सेल ले ली, एक मैक्रोफेज ले ली, इस पे CD4 पॉजिटिव रिसेप्टर है, तो जैसे वायरस इन्फेक्ट कर गया, होस्ट सेल पे पहले बाइंड करेगा इसी रिसेप्टर पे बाय द हेल्प ऑफ GP120. तो जब हम देखते हैं कि जो ये सेल्स होती है, फॉर एग्जांपल मैक्रोफेज, इन सेल्स पे होते इन CD4 पॉजिटिव रिसेप्टर्स के अलावा कुछ को रिसेप्टर्स होते हैं. दे हैव अदर टाइप्स ऑफ रिसेप्टर्स which are actually co-receptors. So suppose this is the co-receptor. This is the co-receptor. ये को रिसेप्टर है जो पाया जाता है इन सेल्स पे. तो एक्चुअली these co-receptors are mean for binding the chemokines. These are mean for binding the chemokines. तो ये chemokines के लिए होते हैं एक्चुअली. तो these are actually the co-receptors. So जो मैक्रोफेज पे यहाँ पे जो co-receptor है that is called CCR5. Very important. CCR5. CCR5. So the co-receptor which is found in this macrophage, this is called CCR5. What does it mean? It is chemokine receptor. Sorry, 5. I wrote here 4. It's 5 actually. Chemokine receptor 5. So this is chemokine receptor 5. So this is the receptor for chemokines. Chemicals produced by the cells, and then these chemicals bind with these receptors. It's actually for signaling process. Now what actually happens in this case of virus, this virus takes the advantage of this co-receptor. It just takes the advantage. What it does actually, as soon as this GP120 binds with the this CD4 positive receptor, this CD4 positive receptor, there is change, conformational change. One more side gets activated on the GP120. तो जैसे ये बाइंड करता है GP120 CD4 पॉजिटिव रिसेप्टर, और एक साइड इसकी एक्टिव हो जाती है. जो साइड बाइंड करती है, एडवांटेज लेती है, अपॉर्चुनिटी लेती है और बाइंड करती है इस को रिसेप्टर के साथ, which is CCR5. So this is actually a co-receptor. This is a co-receptor. So CCR5 पे अब ये bind करेगा another active site which has been activated after binding of the GP120 with CD4 positive. So this active site gets activated. It's just like the hand which is in my pocket. When first this GP120 first primary attachment is done with the CD4 positive cell, the active site is in my pocket. When there is primary attachment, this site gets activated and binds with the co-receptor of this macrophage, that is CCR5. When it binds with this, what actually happens? This GP41, it just changes, it moves to a side, and slips to a side and hooks the membrane. What it does, this slips to side and hooks the membrane. It will try to bind the membrane, hook the membrane. So this GP41 will hook the membrane, or we can say it will fuse the membrane. It's very important here. So we can say when the active site binds with the co-receptor CCR5, there is change in this G41. It just moves to the side, slips to the side and hooks the membrane of the host cell and tries to fuse it, tries to pull it towards itself and fuses the two membranes. This is lipid membrane outside, lipid bubble, and this is lipid bilayer lipid membrane, and these two membranes will fuse. So this GP41 is a fusion molecule. That means इसका मतलब हमें याद रखना चाहिए यहाँ पे. It's worth to note that GP41 is a fusion molecule. It just fuses the membranes together. GP120 is the primary attachment protein, glycoprotein. So when the two membranes get fused, it's like that. Now they have fused together. This is the virus and this is the macrophage. This is the macrophage and this is the virus. When the two membranes will fuse, they get dissolved, the membranes get dissolved, and the virus will enter into the host cell. So like this one, the virus will enter into the host cell. This is how the virus makes entry into the host cell by the use of these two important proteins, GP41 and GP120 and GP41. So this is how it makes entry into the host cell. So through this interaction, the virus is able to enter into the host cell. Once it enters into the host cell, जैसे ही सेल के अंदर एंट्री कर देता है, first of all what actually happens, in the proteases which are found in the cytoplasm of this macrophage, the proteases which are found in the cytoplasm of this macrophage, they will dissolve this capsid. They will try to dissolve this capsid, and the genetic material is now there. What actually it has, it has, we can say this, when this virus makes entry into the cell, it has three enzymes with it. That's reverse transcriptase, integrase, and protease. Now, by the help of, we already discussed that by the help of reverse transcriptase, it will change the RNA into DNA. This is actually this is the reverse transcription by the help of reverse transcriptase. Then this DNA is integrated into rDNA. This is integrated into rDNA by integrase. We know it all also in the previous lecture. Then this will form mRNA, genomic mRNA, and messenger RNA. This messenger RNA will produce the proteins, viral proteins. Long viral proteins will be formed, and these viral proteins, polyproteins, are cut into smaller proteins by the help of enzyme proteases. So then these smaller proteins with this genomic RNA will bind together and form the new viruses, and they will now fuse with the membrane. When new viruses are formed here, we will show it here. When new viral proteins are formed here, this will bind and they will fuse with this membrane. This membrane will evaginate and come out like this in a lipid bubble. Another virus will be there, which are just like the new viruses. So this is how the virus infects the cells and then makes its copies and then comes out through exocytosis, and new viral particles are produced. These viral particles again infect the new cells, and this continues produces billions and billions of cells inside the host cell and infects it. So we now see how it infects the cells. Likewise, it then infects the helper T cells. The helper T cells here have another co-receptor. Helper T cells, as macrophages have CCR5, helper T cells have co-receptor, which is CXCR4. So this is very important again. So helper T cells have another co-receptor, that's CXCR4. These initially try to love with this macrophage, then after many generations they are in a position to bind with this co-receptor of the helper T cells. That's why initially they will bind with the macrophage and they will not try to bind with helper T cells. Then after many generations they will have this ability to bind with the another co-receptor which is found on the helper T cells and infect these cells and start to destroy helper T cells. So this is how the virus makes entry into the host cell. Now let's see this virus. Actually, there are people, we must know that there are people in the world, in some parts of the world, which actually have the virus. If they get infected with HIV, but they do not develop AIDS, they do not have AIDS. If they have infected with the virus and they live a normal life, and they do not, after doing the tests with their blood samples, they do not have AIDS like symptoms, or we can say they do not have developed AIDS. What's actually the reason that these people do not have AIDS? There is very important thing worth to note, that is those people, they are found, they are about 1% of Northern European descendants. This is 1% of the population of Northern European descendants. These people, when they get infected, they do not develop AIDS-like situation, AIDS-like condition. Although they have HIV in their body, but they will not have AIDS. What is actually the reason? Because those people have mutations in these genes. It's very important. These people have mutated genes of these mutated genes which will produce GP120 and this is about the virus. Those people will have mutations on genes for CCR5. So they have mutation in the genes for CCR5 because it's very important receptor, co-receptor, due to which the virus is able to fuse, enter into the cell. So when they have mutation with this gene, their cells won't produce this CCR5. That means they are homozygous. They could be homozygous for CCR5 gene. So when their genes are defective, they have defective genes for CCR5. Defective genes for CCR5. So the 1% of the population of the Northern European descendants, they have an upper hand for this virus because they do not have the functional genes, copies of the gene which will normally produce the CCR5. They have the defective copies of the genes. They could be homozygous. They are not able to produce. The protein fully, which is very important for binding of the virus and making it into the host cell. So these people will not develop AIDS because of defective genes. They are homologous for both these genes, from maternal as well as paternal. So these people do not develop AIDS because of the natural immunity. They have natural immunity for this virus. So we can say what actually means, if a person has HIV, it does not mean that it has AIDS. As we know here, a person having HIV does not mean that the person will have AIDS. This HIV actually is, we must know another point which is very important. This is a lentivirus. HIV is a lentivirus. It's a lentivirus. Lentivirus are those viruses who have very long incubation period. They have very long incubation period. So lentivirus are very dangerous because they take long time. They have a long incubation period, after which they can show the symptoms. This virus, after getting infected, it will remain in silent. It will do not show symptoms up to 7 to 8 years, or sometimes 10 years. So we can say it will take about 7 to 8 years to fully grow into AIDS. So when a person gets infected with HIV, he will be asymptomatic. First he will develop some in the acute first stage of acute infection. He will develop some flu-like symptoms like fever, sore throat, body ache. Then the virus will remain in a latency period. It will remain for 7 to 8 to 10 years without any symptom, asymptomatic. The person will live a normal life for 7 years. After that, 7 or 8 or 10 years, the symptoms will start, chronic stage will develop, and that is the third stage, it will develop the AIDS-like situation, AIDS-like opportunistic, it will develop the immunosuppression, opportunistic infections, neurological dysfunctions, and secondary neoplasia, which will develop into a whole situation called AIDS. So it will take a lot of time, 7 to 8 or 10 years, which is that's why it is a very dangerous virus, which is called a lentivirus. So HIV is a lentivirus. It takes long incubation time, incubation period for symptoms to occur. So now in the next lecture we will talk about how this virus gets transmitted, and how this virus came into existence, and what are the treatments for this virus. We will deal about these things in the next video. Subscribe and like our YouTube channel for the latest new videos, so that you can get acquainted with all the things which we are discussing on these things like HIV and AIDS. Thanks for watching.